The colonic microbiome is that collection of microbes in the colon which has the densest concentration of microbes of anywhere on the body, estimated to be on the order of 10 to the 12, 10 to the 13 organisms per gram of stool, which as we understand today is predominantly bacteria. So I'm an infectious disease specialist who started in the field of diarrheal disease and developed an interest in diarrheal disease in a refugee camp. So came back from working in a refugee camp and studied for about a decade a bacterium called enterotoxigenic Bacteroides fragilis that caused diarrhea in children. In the course of that study, we recognized that the mechanisms by which the organism was acting and the toxin that it made kept intersecting with oncogenic pathways. And at the end of that time, it dawned on me that as the microbiome was starting to be discussed, that we needed to ask the question about whether that bacterium, if it was embedded in that mass of bacteria, might influence carcinogenesis in the colon. So we did a mouse study. It was dramatically positive, and that launched us into understanding the mechanisms of that bacterium in mice. But as a physician, it of course led me back to the clinic to try to understand how the microbiota may be contributing to colon cancer. So we are working on overall just trying to look in as open a way as possible at how the bacterial communities in the colon may contribute to colon cancer pathogenesis. And on one hand, we have this very striking community effect where there's biofilm formation, which are dense clusters of bacteria contacting the epithelial cells, changing the biology of the epithelial cells that cluster to the right colon. And on the other hand, we're finding that particular bacterium that have been described in elegant murine studies by ourselves and others that are oncogenic in mice are also present in excess in these patients. And so our goal is to try to understand this puzzle and how these different factors may interrelate, how they may be causing disease, and how we can translate this information into better tests to understand who's at risk for colon cancer. Our study was cross-sectional, so understanding the earlier stages of that is not possible. So what we think is important to do is to follow a set of patients over time to understand if those who have a biofilm in their colon have changes over time consistent with developing processes consistent with carcinogenesis in the colon. So really looking in the healthy host, where a small subset of people will have biofilms, and understanding if there's evidence of progression of the biofilms, of the changes in the biology of the epithelium, in the development of tumors over time, to understand if it is a measure that we could use to predict a patient at risk. Well, half of my life is devoted to trying to understand more precisely mechanisms of colon carcinogenesis using a mouse model and a particular bacterium or different bacterium that have been associated with colon cancer. And the other part is to really work with our populations who are at risk for colon cancer or have colon cancer to try to understand more about biofilm formation and these different bacteria in those hosts. So we did a study a few years ago with Dr. Maria Oliva-Hemker, who's the head of our division of pediatric gastroenterology, where we were able to collect stools from 100 children with inflammatory bowel disease and 100 children without inflammatory bowel disease. And we're in the process of looking closely at these bacterial populations in those two groups, since IBD is well known to have an increased risk of colon cancer. So individuals with inflammatory bowel disease is another very important patient population to try to understand these interactions, again, because actually the disease can be very hard to diagnose in those populations because it's not sometimes as obvious as in a patient who doesn't have inflammatory bowel disease. Well, at this point, we don't have a message that translates to action in the clinic. However, I think that clinicians should keep an eye out, as a lot of people are working in this field to try to improve our diagnostics for the prevention of colon cancer.